The overall aim of the following experiment is to determine if a potentially heterogeneous sample of biomolecules contains nucleic acid and/or reducing sugars, and, if so, distinguish between RNA and DNA based on the differing reactivity of their sugar moieties. This is achieved by first applying the Benedict assay to determine if the biomolecular mixture contains free reducing sugars. As a second step, Biles-Orsenol assay is used, which establishes whether or not any sugar components contain pentose rings. Next, Dish's diphenylamine reagent is used to determine whether the pentose sugar is a deoxyribose, as in DNA, or not, as in RNA. The results show the type of biopolymer based on readily distinguished color products formed by the differential reactivities of sugar-based components in the sample, which can be analyzed against standard curves using spectrophotometric measurements.
The main advantage of this technique over existing methods such as electrophoresis using fluorescent dyes, Pico Green, or Cyber Gold is that there is no limitation based on size, charge, or sugar component, and it is efficient in time and cost.
To assay for reducing sugars, prepare a suitable volume of 6x Benedict's reagent composed of 940 millimolar anhydrous sodium carbonate, 588 millimolar sodium citrate dihydrate, and 68 millimolar copper-2 sulfate pentahydrate. For each sample to be assayed, add 100 microliters of the 6x Benedict's reagent to a 1.5